Uh, Dr. Shah, if you're unable to share your with your computer, you can send me and I can handle the slides for you.
Okay, I can, I'm trying to see if I can, hold on.
Yeah, sure.
Yeah, yeah, I found it.
Yes, it's perfectly visible.
Yeah, can you see that?
Yeah,
Okay, okay,
sure.
let's begin. Um, so thank you very much, Papana and King Edward Medical. What do you call it, usually? The
Uh,
Academic Council?
no, no, it's Chem Psych. We have made this psychiatry interest group for the medical students
Okay,
primarily.
okay, okay. So thank you for arranging this talk, because one of the main reasons why I wanted to do this is because psychopharmacology has become so complex and difficult to understand that it is almost impossible to do a good job in psychopharmacological treatment for our patients if you don't understand the basic psychopharmacology, and in that sense the basic pharmacology. And that is one of the reason, the interest in psychopharmacology is declining. I have seen that among residents and other trainees. So one of the way we can deal with this declining trends in interest in psychopharmacology is to do our presentations, make them more interesting, more understandable. And for that, I think the difficult foundation has to be laid down before we can do that. For that reason, I just wanted to discuss the nuts and bolts, which is the basics of psychopharmacology, so that you have a solid foundation on which you can build on. So that is the main goal behind this. I may not be able to finish this lecture today because there are a lot of slides, but I think my plan is that I have four lectures in this series. I was planning to move on to antidepressants once we are done with this, so that I think that may be most helpful, because that is probably the most frequently used psychotropic class in psychiatry. So maybe we can build on this and then move on to their specific psychopharmacology. So let's start. No, so the theory of drug action has been not new. In 1890, somebody came up with this idea of lock and key hypothesis. And it's pretty, I think for those times, it's pretty neat that this scientist, Mr. Fisher, could actually understand that the neurotransmitters um, actually need to fit the receptors to create an action. And so that actually was the first kind of hypothesis related to drug action. But I think it is not a perfect hypothesis by any means. As you can see that um, only the key which fits the lock can open the lock, right? But receptors is not a monolithic concept. Receptors are of different types. Drugs and neurotransmitters can have different effect on receptors. Some can be antagonists, some can be agonists, some can be partial agonists, some can be inverse agonist, and so forth. And we'll talk about that. But I think the lock and key hypothesis does not explain the partial, for example, opening of the lock versus a complete opening or a lack of opening. So although this started the debate on the drug action hypothesis, this is not by any means a perfect hypothesis. So I was just thinking about this, and one of the reason why I came with the nuts and bolts is because probably nuts and bolts provide a little bit better hypothesis for drug action. So if you can imagine that if there is a nut which a bolt goes into the nut and fits it perfectly, that is like a lock and key. But if you let me hypothesize, maybe it's a far-fetched idea, but if you let me hypothesize, on the right side, you see that there are bolts with they don't have grooves all the way. So that can actually partially can do a partial function, if you can understand me. Also, I think there are some nuts which actually may fit loosely to the bolt. And that actually creates the other interesting concept in psychopharmacology. So I don't want to spend a lot of time, but this is kind of interesting that nuts and bolts hypothesis may be a little bit better than the lock and key. So what are the targets? Let's start with the real discussion here. What are the targets for psychopharmacological agents? So this is the core of this presentation, that we are going to talk about these things. So please, if you don't understand, please write down the question and you can ask me later. I wish this could be a more interactive session, but unfortunately, because of the virtual presentation, I think we are limited. Because I love to have interactive sessions where we can the audience can ask questions, or even I can ask questions. That what is the understanding of the audience? So as you can see, the number one bullet actually talks about transporter proteins. You may have heard about transporter proteins. There are some transporter proteins which are also known as reuptake inhibitors. For example, the most frequently used class of antidepressants, which is called SSRIs, actually is the selective transporter inhibitor for serotonin, and it is called selective serotonin reuptake inhibitor. The reuptake inhibition has been recently is now better termed as transporter protein, because that's actually what they are. So this is an extremely important target, molecular target for psychopharmacological agents, because 1/3 of all psychotropic actions, medications action, is explained by transporter proteins. So the transporter proteins are, again, is not, to make life more complex. As we move through this lecture, you will see the complexities of layers of complexities one after the other. So the one problem is that the transporter proteins are not actually a monodimensional concept. And there are membrane transporters. For example, the neurotransmitter transporters, such as serotonin transporters, or norepinephrine transporter, or dopamine transporter, or GABA transporter, or glutamate transporter. All those different neurotransmitters have those transporter proteins. But there are other transporters which are much less known than the neurotransmitter transporters. For example, P-glycoprotein. I don't have time to discuss in detail, but P-glycoprotein is a very, very important transporter, which is energy dependent and actually is responsible to extrude, eliminate the drug from within the cell, outside the cell, because the cells, because our body considers drugs as toxins. So that is kind of a defensive mechanism, which is, as there are many other defensive mechanisms we'll discuss through this presentation. So that is also important. Then there are transporter proteins in the vesicles, which are in the presynaptic neuron, and actually they actually facilitate the reuptake of the ingredients to make neurotransmitter, whatever the vesicle is for. For example, serotonergic vesicle will actually uptake tryptophan and the enzymes which are required to manufacture serotonin. So these transporters are very important too, because if there is a problem with these vesicular transporters, the vesicle will not be formed, and there will be deficiency of the neurotransmitters. So this is a very important concept to understand. And then we moved on to G protein coupled receptors. So the G protein coupled receptors is also responsible for about 1/3 of actions of all the psychotropic medications. And these are the majority of receptors which we have. And as we move on, we will know which one are those. These GPCRs, which is G protein coupled receptors, actually can do so many different things. As you can see, they can regulate enzymes, they can regulate ion channels, they can regulate transporter proteins. So they have all different types of action. Some people think that the G protein coupled receptors, which are also known as metabotropic receptors, actually, why do they act on ion channels? Because for that we have ionotropic receptors. But this is the complexity of our brain, that even the G protein coupled receptors can also do work on the ion channels. So the next one is the ligand-gated ion channels, which I was just talking about. They are also called ionotropic receptors, and they actually depend upon the passage of some electrolytes, mainly calcium, sodium, and chloride, to create an action potential, which results in cascades of reaction and the transmission of the neuronal impulse. Okay. So the next one is voltage sensitive ion channels. So they are ligand gated, and then they are voltage sensitive. They don't require the electrolytes, but the sodium actually participates in majority of those. And when the sodium enters, it changes action potential, results in action. So they are similar, but the ligand gated can be affected by the drugs. By the voltage sensitive can only be affected by sodium. So that is the main difference. Okay. Then there are enzymes. So I think we need to actually elaborate on this, because a lot of people think that the psychopharmacological agents usually don't work on enzymes. They do actually. They attach to the enzymes, and there are hundreds of actions which psychopharmacological agents can perform on enzymes. Just for example, one of the most well understood effect of drugs on enzymes is the cytochrome P450 enzyme inhibition by the selective serotonin reuptake inhibitors, SSRIs, and that is a well-known thing. But there are enzymes which actually metabolize neurotransmitters as well, and those are extremely important, because increased or decreased activity of these enzymes, which metabolize neurotransmitter systems, can actually change the whole brain chemistry. So you can imagine how these things are, all of these things are important. And then there are nuclear receptors. And of course, there's always unknown in brain. There's a lot we don't understand, right? So let's move to the next slide. And this slide is extremely important, because if you understand this, you will understand the basic concepts in where are the molecular targets of pharmacological agents, not just the psychopharmacological, but a lot of these targets are available, some of them at least throughout the body. So don't limit it to psychopharmacology, but since we are talking about brain, this is the concept I wanted to give you. So basically, these are all the well-known 7 molecular targets where drugs can go and affect the systems. So the number one is, and let me clarify, the drugs are not approved for all of these, but there are at least 4 or 5 on which I can give you some practical and clinical examples. Okay, so number one is, you see in this cartoon that the neurotransmitter molecules, they are synthesized from the precursors, as I was just telling you earlier, under the influence of enzymes. So what is that? You can see the ingredients are going into the vesicle, and then the vesicle is formed around these ingredients, and then that's a vesicle which actually what. But a lot of people ask, what is the reason why there are vesicles? I think the main reason for vesicles is protective, because if the neurotransmitters were out there in the open, in the presynaptic neuron, they will be eaten up by the enzymes, monoamine oxidase enzymes, which degrade them immediately. So that is the reason. So there has to be a protective mechanism. So that is the reason why their vesicles are there. So that is the first thing which is important to understand in psychopharmacology. And then the second one is the neurotransmitter molecules are stored in vesicles, which I just told you. So these are stored for protection. And the number 3 is the neurotransmitter molecule, then they are leaked from their vesicles, are destroyed by the enzymes. So, so if there are too many vesicles, and if for recycling purpose within the presynaptic neuron, I think some of the vesicles lose the membrane around the vesicular membrane loses the integrity and can deintegrate and release the neurotransmitter to be metabolized by the enzymes. So that can happen too. Another thing which can happen is that there are reuptake pumps or the transporter proteins, as I was just telling you, that actually can block the entry of the ingredients or the products which are required to manufacture a neurotransmitter, and then that can result in deficiency of that particular neurotransmitter. And there are some great examples of those. I will talk about those because there are some recently approved drugs by FDA which actually use that mechanism. And then the fourth one is that the vesicles fuse to the perisynaptic membrane, and then they are released into the synapse, which is also known as synaptic cleft, which is the small narrow area between the perisynaptic neuron and the postsynaptic neuron. And this neurotransmitter is now free to act on both presynaptic receptors and the postsynaptic receptors. And those are also relating of which neurotransmitter you're talking about. There are many, many of those. So add further complexity. And those are the real molecular targets for a lot of psychotropic drugs, as you can imagine. And then we talked about the fifth point. And then let's go to the sixth one. The released neurotransmitter molecules bind to the postsynaptic receptor. And that the number 7 is that when the neurotransmitter is available in the synaptic cleft, there are enzymes such as catechol-O-methyltransferase, which actually can metabolize the excessive neurotransmitter system, in this case biogenic amines, because they are metabolized by COMT, which is catechol-O-methyltransferase, for them to be recycled, or for them to prevent the excessive activity of neurotransmitter, another protective mechanism that God has given us in the brain system. So these are some of the main sites which are extremely important in psychopharmacology. Let's move on. Now the question, how do the pharmacological agents affect them? So this is extremely interesting. Another slide which will take some time. So this will require your attention, because I think if you understand this, you will again understand the molecular targets, potential molecular targets we just talked about in the last slide. So when you look at this, you see that the number 1, the drug serving as precursor, and AGO stands for agonist. So for example, the precursor for dopamine is L-DOPA. And you might know that because the L-DOPA is used in Parkinson's disease, but it is not used as an L-DOPA. It is combined with carbidopa, which actually prevents the peripheral decarboxylation of L-DOPA, because it can readily be metabolized by the enzymes, as we were just discussing. So the prevention of peripheral metabolism of L-DOPA makes it possible for the adequate amounts of dopamine to go into the brain, cross the blood-brain barrier, and go into the substantia nigra, where there is a deficiency of dopamine, and that can improve the symptoms of Parkinson's. So that is a good example of a drug which a carbidopa and L-DOPA combination is called Sinemet, if you remember from your pharmacology, right? So that is a good example of how a precursor neurotransmitter actually is made functionally available in the brain. So that is first. The next one is the drug prevents storage of neurotransmitter in the vesicles, and that is antagonist. Because remember, if a drug prevents the entry of the ingredients to manufacture neurotransmitter, there will be a deficiency of that neurotransmitter, right? And that is the antagonist effect, as opposed to the first one, which I was talking about, which was an agonist effect for dopamine, right? So when you use a drug which prevents the storage of these neurotransmitter in the vesicles, and they deplete those neurons. Let me rephrase, because this is not the blockade of the transporter vesicular transporter protein. It is actually the depletion of the neuron. I'll come back to that later on. Okay. So I was just self-correcting. So Reserpine, which is a very old agent known to us, actually was one of the first psychopharmacological, I would not call it a psychopharmacological agent because it was a disaster. It was used to treat psychosis and affective disorders. But the problem was that the improvement lasted for seconds or minutes. And then the reserpine depleted the neurons of the neurotransmitter, which are essential. Some level of their activity is absolutely essential for normal brain function. Also, the patient used to crash. So that was the problem with reserpine. The next item on the left side is a drug actually stimulates release of a neurotransmitter. Very important mechanism, and it is an agonist, of course. Why? Because a drug is going to the presynaptic neuron, and through various mechanisms is facilitating the release of the neurotransmitter from the presynaptic neuron. And a good example is acetylcholine, which actually is caused by the black widow spider venom, as the example is given here. But there are better examples. For example, amphetamines can enhance the release of neurotransmitter systems, and also actually the methamphetamine can block the release, can increase the release of neurotransmitter from the presynaptic neuron. At the same time, can also block the reuptake pump. So it results in huge increase for dopamine in the synaptic cleft, which is a problem, as obviously you can see. Then the next one is the a drug inhibiting the release of neurotransmitter from the presynaptic neuron, which is an antagonist effect as opposed to the other one I just talked about. And botulinum toxin is a good example in relation to acetylcholine, where it actually inhibits the release of acetylcholine from the presynaptic neuron, and results in problems, which actually is being used therapeutically these days. There is FDA has approved it for the treatment of several indications, and actually in psychiatry, there are a lot of studies, actually multiple studies, which looked at the use of botulinum toxin injections in the forehead region, and that has been shown to help depression. If you're interested in that, you can go and we did a review on this, on all the studies which are available. Very interesting ideas on this. And then the next one is the drug stimulating the the postsynaptic receptors, which is also agonist effect because they are stimulating the postsynaptic receptors. And great examples of this is, I'll give you example of SSRIs, which are actually they block the reuptake pump. But after the reuptake pump is blocked, increased serotonin is available in the synaptic cleft, which is available to act as an agonist on all the postsynaptic serotonergic receptors. For example, nicotine, muscarine in acetylcholine system are other examples of how, and there are some drugs actually which directly go and stimulate the postsynaptic receptors, such as Buspar, which actually not only activates the presynaptic receptors, but also the postsynaptic receptors. 5-HT1A, which is a subtype of serotonin receptor, they can do that. Okay, there are drugs like metabolite of trazodone, which actually can go and activate a specific type of serotonin receptors, which is 5-HT2C. And we don't have time to discuss all that, but that is very interesting. So let's move to the last one on the left, in the left column. Then that is when a drug blocks postsynaptic receptor, which is obviously an antagonist effect. Well, all these examples are with regards to acetylcholine. I need to actually make a slide showing other neurotransmitters, but I didn't have enough time to do this. But just to give you an example, that there can be drugs which actually can block the postsynaptic receptors, and there are a lot of them. The atypical antipsychotics block 5-HT2A receptor, which is a postsynaptic receptor. They also block many other, depending upon the type of atypical you're talking about. So that is a good example of those drugs. Then moving on the right side, there are drugs which actually inactivate the synthetic enzyme, which results in inhibition of synthesis of neurotransmitter, and they are obviously antagonist. For example, you give PCPA, which actually can block the synthesis of serotonin. And then the next one is a drug stimulates autoreceptors. What are autoreceptors? Very, very interesting concept in psychopharmacology, because if you don't understand this, it will be difficult for you to understand the mechanism of action of so many drugs, but also the basic neuropsychopharmacology will be difficult to comprehend. So basically, these autoreceptors actually uh, they are situated in the perisynaptic neuron. They are not in the postsynaptic neuron. They're almost always present on the presynaptic neuron. And why they are present there? Another God's measure to protect brain from excessive activities of neurotransmitters in our brains. So the autoreceptors actually get activated when a certain neurotransmitter is released in the synaptic cleft, and that neurotransmitter actually stimulates these presynaptic autoreceptors in an effort to regulate the excessive release of the neurotransmitter from the presynaptic neuron, so that they can actually avoid the excessive and toxic availability of a certain neurotransmitter. For example, dopamine. Excessive release of dopamine can be toxic, like other neurotransmitter systems. Anything in excess will be bad, right? So this is another mechanism which is extremely important. And one of the point which I don't want to forget, just wanted to make it now, is that a lot of you would know Eriprisol, which is an atypical antipsychotic, should not be classified under atypical antipsychotic because it is not a second generation. Actually, it is a third generation antipsychotic drug. But don't say that in the exam because you will fail. That's just my effort to make you understand better. So this aripiprazole is actually unique. This is the first kind of partial agonist in in terms of antipsychotic medications. And so being a partial agonist, and we actually can refer that this discussion when we are talking about partial agonist, but that's a very important concept. Coming back to the autoreceptors, the autoreceptors example, the reason I was bringing aripiprazole is because it's also acting on at some level at the autoreceptor. Now, talking about a very common example of an autoreceptor stimulator is clonidine. Clonidine acts on the alpha-2 receptors, which are present in the presynaptic neuron. And alpha-2 receptors, when they are activated, they put brakes on the excessive release of norepinephrine, which is again a protective mechanism. Similarly, BOOSPAR, which is a partial agonist for 5-HT1A autoreceptors in the presynaptic neuron, can also check and regulate the excessive release of serotonin and so forth. Now, the next item on this one is the in the right column, number 9. When a drug blocks autoreceptors, it can actually increase the release of neurotransmitter, which is actually an agonist effect. So if you reverse, if somebody, rather than stimulating the autoreceptor, which I just gave you some examples of, if a drug blocks the autoreceptor, then it will increase in the excessive release of that neurotransmitter. For example, clonidine is an agonist, and when it activates I think this is something which is wrong in the slide, because clonidine is an agonist and it should reduce the norepinephrine, not increase it. So let me give you an example of autoreceptor blocker. Propranolol, which is a beta blocker, is a 5-HT1A blocker. 5-HT1A is an autoreceptor in the serotonergic system, and propranolol actually blocks that 5-HT1A receptor and can enhance the release of norepinephrine from the presynaptic neuron. That has been actually studied in at least 3 studies in which giving a beta blocker actually speedifies or shorten the response for antidepressant response from antidepressants when a beta blocker just such as Pindalol, which is another beta blocker, is combined with SSRIs or other antidepressants. So that is interesting. And then the next one is the drug, when a drug blocks reuptake. I don't have to give you example of this because SSRIs, we just talked about that. And then the last one is when a drug inactivates an enzyme which metabolizes a neurotransmitter, such as anticholinesterase medications used in Alzheimer's. That's a good example of that. Okay, so let's move to the next. So there are different type of chemical messengers. They are not just the neurotransmitters. They can be endocrine hormones. They can be neuroendocrine. They are called neurohormones. They can be actually chemical messages can be carried by the paracrine signaling as well as autocrine signaling. So again, further complexity that the neurotransmitters, if they didn't make our life difficult, on top of that, we have hundreds and thousands of chemical messengers in the brain, which can actually further complicate the brain function. Now, there are three types of neurotransmissions in the brain. One is on an anatomical basis, which is actually primarily mediated by the neural tracts, and they are developmental, as we can imagine. They are electrical, they are action potential based, and then they're chemical. Most of the neurotransmission is actually a combination of electrochemical neurotransmission. For the sake of time, let's move on. So, so there are some major differences between the parts of a neuron, the different parts of the neuron. There's a cell body, there are axons, there are dendrites, and we need to understand that, and the reason why I'm talking about this very briefly is because first, we don't have time. The second is, and the reason why I still want to briefly talk about this is that when you know the location of a receptor, based on whether it's in the axons or dendrites, actually it makes a hell lot of a difference in the activity of that drug. That is why it is important to know. So actually everybody knows this from the basic neurology, that the axons are efferent and they actually take away information from the cell body. Dendrites are afferent. I'm not going to spend a lot of time. Axons, one very characteristic feature of axons, that they have myelin sheaths, which actually are insulating these axons from the confounding effects of their environment, because they are long tracts and they have this white matter myelin sheaths surrounding them. So there are no electrical mishaps which occur during the transmission of an impulse. Dendrites are not myelinated, very important characterization, and the dendrites can also form a major part of the gray matter, and the axons can form a major part of the white matter in the brain. Now synapse, a very brief discussion of synapse. There are three different types of synapses. Commonly, there are many, there are others too, but these are the ones which, to avoid complexity, these are the best understood. So they can be, a synapse can be between an axon and another axon. It is called exoaxonal. And you can see the diagram on the right side. You can see that where is the exoaxonal. They can be exodendritic, which means the synapse is between axon and of one neuron and between the dendrite of another neuron. They can be exosomatic. The synapse can be between an axon and the cell body of a neuron. So these are important synapses. And each synapse actually, again, in relation to the location of the receptors, can make a big difference. So different types of receptors. The receptors can be excitatory and or inhibitory, as we all know. They can be altered by the type of neurotransmitter, or the type of ion channel which is affected, as you will see down the road. They can be autoreceptors and they can be heteroreceptors. We just talked about autoreceptors, right? These are the receptors which provide the negative feedback loop for the neurotransmitters so that their excessive release can be monitored. Heteroreceptors is a very interesting concept. This is actually hetero means different. For example, if a serotonergic neurons is communicating with a non-serotonergic neuron, the serotonin will go bind to the certain receptor on the other neurotransmitter neuron, and it will bring in the changes which are very interesting. So those are called heteroreceptors. For example, norepinephrine effect on serotonin or vice versa, or so many different neurotransmitters. So those are important. And they are extremely important in the mechanism of action of mirtazapine, which is an antidepressant. I don't have time to discuss that fully, but when we talk about antidepressants, that will be helpful. So see how this knowledge, this foundation will help you eventually to understand psychopharmacology. And then last but not the least, the receptors can be ionotropic or metabotropic. Ionotropic when they affect the ion channels, and metabotropic when they are mediated by the G protein coupled receptors, as we discussed earlier. Now, the agonist spectrum. This is a very, very important slide to understand because it will provide you a good concept of what different type of actions a drug can have on a receptor. Or what is the nature of the receptor, either whether it is an agonist or antagonist and so forth. So an agonist. And this example is using the ion channel because it is very difficult to understand the G protein coupled reactions with the concepts of agonist and antagonist. Diagrammatically, it is much more easier to understand using the ion channels. So the agonist on this rainbow on the left spectrum of this, okay, the left side. And then there is partial agonist. Now observe the size of the lumen because that is important. An agonist has a much wider opening in the ion channel as compared to partial agonist, right? Then look at antagonist. Compare that with partial agonist. It is the smallest of the three, right? And then there is another one, which is difficult to understand at times. It's called the inverse agonist. Inverse agonist is the most potent antagonizing mechanism in the brain, because inverse agonist means it's completely opposite of the agonist, and it completely blocks the lumen of ion channel, as you can see. So these are the three major concepts in terms of receptor modification or modulation based on their properties as an agonist versus antagonist versus partial agonist and inverse agonist. So for example, if an agonist binds to an ion channel, and on the left side of this, there's only one diagram here. It is actually shown in the resting state, okay? So there is an absence of agonist. Look what happens when agonist comes. It widens. The pathway for the electrolytes to enter widens. This is ion channel, right? And so when the agonist binds, then there is more passage of that action potential via the passage of more electrolytes, sodium, calcium, whereas chloride actually are inhibitory, but we'll talk about that later. Antagonist, resting state again on the left side. Look at the size of the lumen, and it actually decreases the outflow passage of the electrolyte through the channel. Partial agonist, resting state, and what it does is somewhere in between agonist and antagonist because it is a partial agonist, right? Inverse agonist, it locks down. It's very nicely shown on the extreme right. There is a lock. Inverse agonists are the most potent antagonist of all actions, receptor actions. neurotransmitters. See how the time flies. Already I'm past. So if you want to stop here, and if there are any questions, um, I can do that. But if you want me to move on, we only have 1 hour, right? Usually.
We can have a little bit more than an hour. We are totally okay with it.
Okay, so let me go through the neurotransmitters and then we will stop. Okay.
Sure.
So actually, there are criteria to acknowledge that some chemical is a neurotransmitter, and those are enumerated here. I think the most important one is that it must be synthesized and released from the neurons. That is no brainer, right? It's a neurotransmitter, so obviously we need to have that. And then there's a lot of other characteristics which I'm not going to spend a lot of time, but but there are these criteria, so you will have access to them and you can look at that. So there are some unconventional neurotransmitters as well. For example, the growth factors. For example, the most important one, which I'm sure you must have heard about, is the brain-derived neurotrophic factor. There is a spelling mistake there. Factor, BDNF, and BDNF is actually one of the most interested factor which has been researched on these days, not only in genetics, but also in terms of response to antidepressant medications, because we have seen that if there is a deficiency of BDNF, no antidepressant, almost as far as we know, when the research is available with the specific antidepressants, especially SSRIs, they are not effective. So it is a marker, a biomarker of antidepressant response, that if the SSRIs are not able to increase BDNF, it is highly unlikely that you will have antidepressant response from SSRIs. Nitric oxide is also, it's a gas, but surprisingly, it is a neurotransmitter as well. It is one of the mechanism of action in sildenafil, which is Viagra, is supposed to be mediated by nitric oxide. And anandamide is the marijuana receptor, which is endogenously present and can be affected by cannabinoids use. And then, of course, this is for TSC receptor. It's a ligand for the TSC, tetrahydrocannabinol receptor ligand. Neurotransmitters can be amines. There are many different types, right? They can be amines, they can be pituitary peptides, they can be neuroactive peptides, they can be circulating hormones, they can be hypothalamic releasing hormones, amino acids, gut hormones, neuroactive steroids such as estrogens and androgens and progesterones and allopregnanolone and so forth. So the sky is the limit. This is just mind-boggling that we have so much, and we are trying to do it in 1 hour is impossible. So I'm going to take more time, a little bit more. Peptide transmitters, I don't have to talk about all of them, but there's a huge list of these. And the one, let me see if I can go back. I can, I don't know whether I'll have opportunity to talk about this, but let's see, okay. And then there are opioid, in terms of opioid peptides, we know that they are dynorphins, they are enkephalins, they are endorphins, all different types of opioid peptides, which have a significant effect on brain function. There has been some recent research, actually it's not that recent, we have known this for a little while, that use of opioid agonist actually may help with the antidepressant effects. And there's some studies going on with buprenorphine, which is a partial agonist at opioid mu receptors, right? So there's a lot of exciting stuff happening these days. And then there are peptides which are miscellaneous. For example, one which comes to my mind to briefly mention is orexin. And orexin antagonists are becoming very popular. They are very expensive, but they're becoming very popular in improving sleep. For example, suvorexant and lemborexant, those are the agents which actually are acting on the orexin. Then gases we talked about, then anandamide, which is a lipid neurotransmitter, and then neurokinins and tachykinins, which is kinins, which actually are working in collaboration with some of the other major neurotransmitters, such as substance P works with serotonin, especially in the spinal cord, to modulate the perception of pain, which is important. And then don't forget about purines. Adenosine is one of the best known agent which actually builds up with exercise and helps you sleep better. That is a very important point to note, that people who don't like doing exercise needs to exercise because it increases their adenosine and they have a restful sleep. That is actually confirmed by the research. Two basic kinds of neurotransmitters, excitatory and inhibitory. We'll talk about them. But to the surprise of the audience, look at this. Would you imagine that GABA and glutamate occupy the major part of the neurotransmitter pie in the brain activity? I think, I have spent years in psychopharmacology and imaging research and genetic research in psychiatry. But although I had an idea that GABA is the most abundant inhibitory neurotransmitter in the brain, and glutamate is the most abundant excitatory neurotransmitter in the brain, I didn't have idea that the part of the pie shared by all the major neurotransmitter systems we love to talk about is very small. And as a matter of fact, there are some researchers which actually are saying that the receptors such as dopamine, serotonin, norepinephrine, and nitric oxide are actually the modulators, modifiers, not the actual mediator of the effects in the brain. And GABA and glutamate, and to some extent acetylcholine, which we used to not consider that high in the line, actually is more important than all the others. Actually, it is even broader than combination of all the others. So please remember this. And the reason I'm saying that is because the future research is going towards GABA and glutamate, and rightfully so. We are late, but I think it's happening. Glutamate. Let's talk about the first, which is the most important and most abundant neurotransmitter, not just the excitatory, but more, as you just saw in that pie chart. So this is excitatory, as we discussed. It is actually a byproduct of glucose metabolism in the Krebs cycle. Interesting, huh? It is actually removed by reuptake. So there are glutamate reuptake pumps or transporter proteins which actually perform the duty. And the most interesting difference between glutamate and other neurotransmitter systems is that the glutamate is actually reuptaken by a glial transporter, not a presynaptic or postsynaptic. Very interesting. That tells you how God has created a special apparatus for this neurotransmitter to function. Oh, of course, it can be neurotoxic, but let me tell you, it is also essential. You cannot do brain function without this. So this is a typical cartoon which actually depicts the presentation of a glutamate tripartite system. Why I'm calling it tripartite? Because it is comprised of three things. One is presynaptic neuron, one is postsynaptic neuron, and one is the glial cell, the astrocyte. So see how the astrocyte or the glial cell has the reuptake pumps, which are called excitatory amino acid transporter, EAAT. There are some in the presynaptic neuron, but majority of those actually are in the glial cell, okay, as you can see. Okay, so these are the transporters which reuptake glutamate into the glial cells. They make it into glutamine, convert it into glutamine, and then the glutamine transfers back through another type of excitatory amino acid transporter. It is taken up into the presynaptic neuron, and the glutamine is converted into glutamate, and then earlier, and then released through exocytosis in the synaptic cleft to act on their several glutamate receptors in the postsynaptic neuron. So see how interesting this cycle is. No other neurotransmitter system has this. Excitatory amino acids, let's talk about this. So there are two types of glutamate receptors. One is called the metabotropic receptors, glutamate receptor, and the other one is called the ionotropic glutamate receptor. So the ionotropic glutamate receptors are of three types. See, layer of another complexity. The glutamate receptors are of three types, as you can see in this the cartoon below. There are AMPA receptors, glutamate receptors, which are ionotropic. NMDA, of course, you would know. Most people know NMDA. It's becoming very popular with the ketamine rapid antidepressant onset of effects, because ketamine is an antagonist of NMDA receptor. And then kainate receptor is another one, which is the third type of ionotropic glutamate receptors. We talked about this. One point, that the metabotropic receptors are called as mGlu-R. M is for metabotropic, is for glutamate, and R is for receptor, mGlu-R. So there are multiple types of this, we'll see. Now, this is a little complex slide, but it's very important to understand again. And throughout this presentation, I'll make several points about how the systems in the brain are interacting all the times with each other. And the brain is not a vacuum. It is a soup of chemical in which one chemical is interacting with the other. And this is a perfect example of that look at the non-NMDA receptors and NMDA receptors. So the NMDA receptors causes influx of calcium into the neuron. And then that the calcium actually enters, and it results in cascades of reaction, which results in after repolarization, and results in the genetic information to be translated into formation of new proteins, right? But look at this feedback loop. The repolarization can come back and inhibit magnesium, which actually blocks that. So there is a regulatory mechanism. But not just this, look at the non-NMDA receptors. The sodium channels can also can come back and say, hold on, magnesium, you cannot do this job. Release this, because we need more glutamate action. So it goes back and removes the magnesium blockade. You see how important this interaction is to maintain this balance, what we call as monitoring the activities of each other and keeping a balance, which is the essence of homeostasis and essence of life, right? So this is a glutamate receptor. You should tell me if because the receptors, a lot of slides on these. So if you want me to stop anytime, let me know. We can complete this later, okay?
Sure. You can carry on right now, sir.
Okay. So the ionotropic glutamate receptors, this is the NMDA. And you can see this is a very helpful slide, because it tells you what are the agonist and what are the antagonist, and what is happening with the glycine. So glycine is one of the most important co-agonist factor in glutamate activity receptor, NMDA receptor activity. If glycine does not attach to the site where it is shown in this cartoon, I think we will have extreme deficiency of glutamatergic activity. And you know what that is going to result into? I think a lot of you might know that glutamate is actually is one of the most important neurotransmitter in long-term potentiation of memories, which is important for us to make memories, right? And cognition, cognitive abilities. So that is there. Then the zinc can do a little bit of that part. And then the calcium channel, of course, is ion channel. You can see the magnesium blocks it, right? The red part of the channel in that the red icon or sort of that, right? And then the PCP, phencyclidine, which blocks NMDA receptor also. So you see the agonist, co-agonist, and the antagonist here, right? And the glutamate binds, it opens the channel, calcium goes through, and the results in cascades of reaction. So let's move to the next slide. This is a metabotropic receptor. You see the complexity of this. Now you will know why I said that ionotropic is the simplest receptor. And look at this. So I'm not going to go a whole lot of details with the metabotropic receptor, but see, there are so many steps for these receptors to result in meaningful changes in the protein. So to bring out the changes which we see, one of the changes, of course, is the formation of second messenger systems and third messenger systems and the fourth messenger systems and so forth. GABA uh, is the next one. So I think we should, because I don't want to have an information overload. This is very complex. I don't want. So let's give some time for questions if there are any, and then we'll start with GABA next time. Is that okay, Yushay?
Yeah, that's perfectly fine. That would be
Okay.
better.
Okay.
So, Dr. Shah, there are a few questions. First one was from Dr. Fozia Chima. She was asking that if you can repeat your point related to aripiprazole and autoreceptors. Aripiprazole regards to autoreceptors.
Yeah. So aripiprazole actually has three core mechanisms of action. Primarily, it is a D2 receptor partial agonist, and that actually and a lot of probably would not know that D2 receptor is actually a autoreceptor. A lot of people don't know this. So when you activate D2 receptor, it will decrease the outflow of dopamine from the perisynaptic neuron. But there are also postsynaptic receptors. So this is very complex, but I'm just trying to try to answer the question. So the D2 receptors are autoreceptors, and since it is a partial agonist, which means that it is neither going to block or completely stimulate the D2 receptor in the perisynaptic neuron, which is a blessing. You know why? Because schizophrenia is a paradoxical condition based on the dopamine circuit involvement in the brain. So schizophrenia, the biggest dilemma in schizophrenia for us to treat schizophrenia has been the basic pathophysiological dichotomy in schizophrenia. By that, what I mean is that schizophrenia is primarily because of excessive activity in the mesolimbic tract of the dopamine system in the lower cortical centers. But in the higher cortical centers, in the mesocortical dopaminergic tract, there is deficiency of dopamine activity. So the point is that if you give an antagonist, especially a potent antagonist, it may treat psychosis, but it will further cause problems by further reducing the dopamine activity in the higher cortical centers. And that can be reflected as depression, negative symptoms, cognitive deficits. So that's the issue. So in that sense, partial agonist offers the best at least theoretical model, because it is neither antagonizing nor stimulating fully. So it provides a kind of a moderation, sort of a system stabilizer. And that's what I meant by autoreceptor. But aripiprazole is not just a system stabilizer for dopamine. It is also a system stabilizer for serotonin. Why? Because it is also a partial agonist for 5-HT1A receptor, which are the autoreceptors for serotonin system. So that's where the autoreceptor comes with aripiprazole. Thank you for asking such a good question, because I was not, because of the time, I could not explain this in detail. Mm.
Right. Dr. Shah, there is another question from Dr. Jamal, and he's asking about vortoxetine, the new antidepressant that have been in the market. And he's asking either vortoxetine works by increasing brain-derived neurotrophic factor, and is it common in all antidepressants, or is it something that is particular to a few antidepressants?
Yeah, so are you talking about fluoxetine?
Vortoxetine.
Oh, vertioxetine. Okay.
Yeah.
Okay. Trintellix, right? Yes, vertioxetine is, uh, I can give you a much elaborate answer when we talk about antidepressant, because I love this drug. This actually, vertioxetine is still a patent and very expensive drug, but you cannot believe it, that before vertioxetine came into the market, there was not a single antidepressant which has some positive effects on cognitive function. And this is the only one which actually improves the attention and concentration and vigilance. But it does not improve the other cognitive functions, but just this one. But imagine a depressed patient's attention and concentration improving. How nice is that, right? So vertioxetine, and I cannot, there's a detail of its mechanism. It's a multimodal drug. It has effect on subtype of 5-HT1 receptors, 5-HT1A agonist, 5-HT1B partial agonist, 5-HT1D antagonist. So I cannot go through that because very complex. When we talk about antidepressant, we'll talk about it, okay?
Great.
On top of that, it is also a serotonin reuptake pump blocker, of course.
Okay, so it means vertioxetine is a little bit different from many other antidepressants in the market.
At least at this point in time, I can tell you in terms of cognitive effects, definitely. Why? Because I'm not the one who's saying this. FDA has approved it for improvement in this specific aspect of cognitive function.
Right, right. So, Dr. Shah, just for the people who are right now with us, what should we expect from your lectures in the coming weeks? Like what we are going to cover in them, and so that they know what they are signing up for the next coming weeks.
Yeah, this is unfortunately, this is kind of a bitter pill to swallow because it is a very highly complex topic. But my sincere, my honest feeling is that without understanding this, how can you function as a good psychopharmacologist? There is no way, right? Psychiatrists are doing such a poor job in clinical practice, creating such a bad name for psychiatry. Why? Because first of all, not all of them, but a good number of them are not trained well in psychopharmacology. The translational neuroscience aspect is not there. You don't talk about this. A lot of people will talk about the receptor, but will not tell you what impact it has in terms of clinical sense. I don't do that. I don't like to do that because I want to link this with the clinical. Otherwise, it's useless. So I'm trying to make it as interesting as possible. But I think it's a bitter pill to swallow. You can review this. I think this will be a recording. Try to remember the things which are the major things in this presentation, because believe me, I have done this 1,000 times and I still forget things. This is very complex and I have to review before I do any presentation so I don't make any mistakes, because things are changing. Things are changing rapidly. Now, who would know that MDMA assisted psychotherapy will be the best treatment for PTSD based on the available research we have now, right? Things are changing. Psilocybin, these things are coming out. Buprenorphine has brought a revolution in the treatment of addiction as a aversion therapy, right? Risk aversion. So risk reduction, risk minimization treatment, right? So things are happening so fast. It's difficult for anybody to keep pace with it. I just came out with attending a meeting with American Society of Clinical Psychopharmacology, and you cannot imagine what is happening out there. But the ironies of the situation is that information is not translated to our trainees and medical students. It is really a dilemma for us, and it does not help, because if I cannot motivate you, Yoshe, to think about psychopharmacology and maybe become a researcher, I'm not doing my job. You have to create interest. So I think in future what we'll do is we'll finish with this, and then we'll talk some practical psychopharmacology, starting with antidepressant, and then if we have time, we'd like to do antipsychotics too, and topics of and we can maybe do in few months another lecture series, because I have so much to discuss with you guys.
Thank you so much. Over to you, Dr. Zishan. Dr. Zishan is an attending in child psychiatrist in Rutgers. So, Dr. Zishan, over to you. You wanted to say something.
Yeah, no, thank you so much, Dr. Shad. I think I cannot emphasize enough understanding the basics of psychopharmacology. As a child psychiatrist and as an adult psychiatrist, we have seen so many times that people are just prescribing medicine because it's an antidepressant.
Yeah.
Even as you mentioned, each medicine, either it's a fluoxetine, although they're in the same category, but each medicine is different, practically as compared to other medicine. So I think it's important to highlight, and I think it's important to hear these things, from a psychopharm expert like you, that can emphasize and explain in a very simple and a very, informative way. I would also ask Dr. Shad if you can recommend, some books for our students so that they can read about these things. Because and where they have these more pictures and explanations so that they have more understanding. But a great presentation, and I think very important both for as an applicant level, as a resident level, and as an attending level to understand the basics. So thank you so much.
Thank you, Zishan. Thank you. I think a lot of credit goes to you, Zishan, for bringing a lot of motivation and enthusiasm in Papana. I think we are becoming an entity now, a recognizable entity, and that is and it's very rewarding because you cannot imagine the interaction which I have with you, my cabinet members, my medical students, my trainees, such as Yushe. You cannot imagine the positive effect of that on my motivation to learn more. You cannot underrate that. And let me tell you, people who are trainees and want to learn more, please teach, because there's no other way of learning than teaching. Teach and teach the heck out, uh, because then you will have to study. Otherwise, who cares, right? We are on our cell phones, our laptops. Um, we're losing the touch of social interactions, becoming more socially isolated, precipitated more by COVID. We need interactions. I cannot survive without interactions. Um, what is the use of this knowledge which I'm learning if it confines to myself and don't spread to the people who want to learn? There's no it doesn't even it doesn't do for a little bit of narcissism which all teachers have in them. It doesn't help at all. Yeah. And we need a little bit of narcissism, not pathological. We need, right? We Yeah, all of us have some personality traits, but being obsessive and being a little bit of narcissistic is the essence, is important. Okay, then we'll do it next week then. Thank you, everyone. And if there are any questions, we have the text. If there are burning questions and I was not able to answer them, please let me know. Thank you.
Can I just make a suggestion?
Yeah.
How about if we have this interactive session 1 week and followed by an interactive session on question and answer, what they learned? Because I feel, on WhatsApp, on text, they may ask, but I think having you live,
Yeah.
they have read this material, they have learned, you can share some reading material, you can read, and the next week they can come up with question and ask you directly, so that they can absorb and also, make it more practical. That's just my humble suggestion.
Yeah, so basically, that's a great suggestion. What I was thinking is that you want to break this presentation, then have a question answer session, and then start with the next other slides in this.
That's what I was feeling. Whole session for the presentation, give them the reading to read, and then have them come up with some questions, and you go live by answering their questions.
Okay, so you want to let me confirm, the next session will be the question answer session, right?
That's my suggestion.
Okay.
So, Doctor, just to add a few things here, we have this WhatsApp group where I added you, Dr. Shad, about the people who are, and I'll share that in the main group as well. So you can suggest some reading materials, and then after a week, after reading those materials, we can have a live discussion on that topic where we can have our questions, and we can you can fill the gaps in our knowledge and in our links that we have come across through that reading materials assigned by you in the beginning.
Okay, so I see that there is more interest in rather than a slide presentation, a more interactive discussion.
Yeah, that would be better.
No,
Remember what I told you? Yeah, remember what I told you when I started? I love to have active session when I'm doing slide presentations, but that is not possible. Maybe it is, but I don't know whether we can stop the slides and somebody interrupt and ask the question. So, so what we can do is we can stop the slide presentation at this point, and then next time do a interactive session with whatever slides I've presented and based on the readings, and then go to the next part then after that. Okay.
Yeah,
Yes.
that
Yeah,
Okay.
because it will help Monica in a way, because there is a role of PowerPoint. There is a role of people understanding
Yeah.
this
Yeah.
from an expert, but
Yeah.
but we need to have full session for the interactive. And if we want to make it a little bit more formal, we can just create some MCQs after this presentation. You could create an
I
MCQ.
do have MCQs at the end.
Yeah. So have them. Let's do some, and then we'll have some questions.
Okay.
Yeah.
Okay. Thank you very much. Have a rest of the weekend, and then we'll see you next week.
Thank you so much. Thank
Bye.
you so much. Thank you so much.
Bye.
Okay.